Welcome to Mr. Od's fourth video on inheritance. This time I'll be talking about genetic diversity. Nature loves variety. This is part of the process for evolution. Nature creates an incredible variety of possibilities and then selects which of those are most fortunate to succeed in the competitive nature of life. Meiosis achieves this diversity through two mechanisms: independent assortment and crossing over. Both of these events occur during meiosis 1 in prophase and are mediated through homologous pairs. A key point here, all this diversity will happen entirely in meiosis 1 early on via the movement of the homologous pairs. This variation in the chromosomal nature guarantees that each individual will be unique and offer their own particular set of genes called alleles. Once again, the homologous pairs are a chromosome from each parent. And during prophase to metaphase, the homologous pairs line up independently along a central axis, resulting in a random distribution of genes. Here you can see in the upper image here, the blue chromosomes appear on the left, and in the lower half, in the central portion, the red and blue chromosomes are on the left and a red and blue on the right. We can see via metaphase 1, this second group here will lead, metaphase 1 and metaphase 2, this distribution of these chromosomes will result in completely different assortments of gametes. That is, in the lower half, each gamete has a red and blue chromosome, and in the upper half, the gametes contain only blue chromosomes or red chromosomes, simply by the arrangement of lining up with this incredibly simple example of only two separate homologous pairs lining up. You can imagine what would happen in the instance of humans with 23 homologous pairs lining up. Great variability simply from independent assortment. We will spend more time learning about alleles in the next section, the next chapter. And we recall that each of these homologous chromosomes really contains the same genetic content. And the particular parts of the content that are matching for a particular instruction set on how to build a particular protein is called an allele. A really phenomenally complicated and amazing process occurs that creates great variability called crossing over. It's also known as recombination. It's the same thing. You'll see in the subsequent slide this crossing over is actually a good name. But it creates this rearrangement of genes, DNA sequence, and resulting genetics through this process makes really different chromosomes. And this picture, I think, is incredibly illustrative of the process of recombination or crossing over. You can see here in the screen that sister chromatids, matching chromatids, are adjacent to each other and in fact swap their genetic content, creating completely new and original chromosomes that have never previously existed. Then this recombined chromosome will separate off and form its own completely new gamete, with the remaining sister chromatid possessing the original instructions from one parent off to a different gamete. Perhaps this creates some of the variability that we see in our siblings. In this point where contact is made between the two innermost sister chromatids is called the chiasma, and the plural of chiasma is chiasmata. The attachment point for the microtubules in the centromere is called the kinetochore. It is a kind of a motor attachment. Here is another image related to this process of crossing over. Here you can see just the central sister chromatids will actually exchange genetic material. The outer portion, outer sister chromatids, remain completely intact from one parent or the other. Some practice questions. How do homologous pairs mediate genetic diversity? Via two mechanisms: independent assortment and crossing over. Recombination happens during which stage of meiosis 1? During prophase 1. Are all sister chromatids subject to recombination? No, some chromatids do not cross over. And what do we call different versions of the same gene? They are called alleles. Regarding this second question, it's really in the process of moving to prophase 1 and metaphase 1 where the crossing over occurs. My daily land acknowledgement to the people who were here first. Thank you to all of our local tribes in Puget Sound. Tigwitseed. You've heard a few times that each human has 23 pairs of chromosomes for a total of 46. The 23 pairs are in duplicate. They are composed of 22 regular standard type of chromosomes, which you see matched up in this karyotype here, plus the sex chromosomes of a different nature, the X and Y, the small chromosome, or the X and the X chromosome. They still remain a homologous pair. And this structure here is called a karyotype. The autosomes contain particular types of information on, say, how to build a lung or a kidney and particular cells, but lots of kind of what you might be considered regular genes also exist on the sex chromosomes. Here is the karyotype one more time. At one point, individuals would match the chromosomes, say this yellow pair here and this yellow pair here, or this blue pair with this blue pair, and snip them out and put them on a piece of paper like this. Now, I'm sure this is all done digitally. Here, the interesting part of this particular image is this nucleus in a more natural state with the chromatin more relaxed. You can see that the chromatin exists in kind of neighborhoods. Sometimes the process goes wrong. A failure for the chromosomes to separate correctly, perhaps a poor attachment of the microtubule spindle fibers that don't function, leaving some chromosomes behind, results in a state called aneuploid, built up of an, meaning not, like an as in anaerobic, u, meaning normal, common, or typical, and ploidy related to the number of chromosomes, not usual number of chromosomes, aneuploid. And here are a couple of instances during meiosis 1 or during meiosis 2, resulting in an abnormal total number of chromosomes. Frequently, states of aneuploidy, that is, an unusual number of chromosomes, is lethal, especially in the larger chromosomes. However, the chromosomes 22 or the sex chromosomes or chromosome 21, individuals are actually walking past us on the sidewalk. The most famous version of trisomy is trisomy 21, Down syndrome. Due to a non-disjunction in meiosis, these individuals tend to be a little bit shorter. They have developmental issues, that is, they do not achieve what we might call a fully adult state. This syndrome is highly variable in its presentation, that is, some individuals you might wonder about them, and others would clearly exhibit symptoms of the syndrome, and often individuals will have a webbed neck. On the other hand, individuals with this syndrome are thought to have really wonderful, beautiful personalities and largely enrich the lives of the people surrounding them. Other of these non-disjunction type of syndromes are Klinefelter syndrome with 2 copies of the X syndrome and the Y syndrome, the Y chromosome. The Y chromosome brings maleness with this, so these people appear as males, tend to be shorter in stature, and have some other features that have variable expression. There are probably some people that you've encountered in your life with this syndrome that you would not necessarily recognize. People with Klinefelter syndrome have a hard time having children. Some other kind of maybe the yin and yang complement to Klinefelter syndrome would be the double Y syndrome. These individuals tend to be a little bit taller. They function quite well in life, and however will not achieve the highest levels of mental function. That is, you would not find them running large corporations. Similarly, individuals with the extra X chromosome, the 3X chromosome, individuals tend to be a little bit taller too and would not be running large corporations, but there are a number of these individuals moving past us on the sidewalk in our daily lives that we would not recognize. Individuals that are missing a chromosome, the X chromosome is sufficient to provide 1 copy, enough genetic material for an individual to develop. Individuals missing the second sex chromosome have Turner syndrome. These individuals tend to be shorter in stature, and again would not be running large corporations, but can carry on lives often unrecognized. Here is Turner syndrome. Individuals are often diagnosed with this because they might have some other systemic complaints that are not resolved, and finally somebody suggests a genetic analysis showing a karyotype here with instead of 23 complete pairs of chromosomes, only 21 pairs of chromosomes with the individual X chromosome. Similarly, Klinefelter syndrome, a karyotype is made likely from a blood sample and a week's worth of laboratory work to come up with an image like this, with, in this case, 23 pairs of chromosomes plus an extra. Finally, a really interesting phenomenon called X inactivation. Females have 2 copies of the X chromosome, which could cause some challenge in expression. However, at some point early on in the development of the individual, certain cells turn off 1 of the 2 X chromosomes, but not every cell turns off the same X chromosome. These cells develop on in kind of regional or geographic populations, resulting in the calico cat here with regions of clear orange or clear black. I was visiting one of my longtime friends and noticed that her cat, Tobiko, also exhibits this kind of mosaic pattern. The X inactivation and this mosaic pattern only occurs in females because the males have only 1 operational X chromosome. and finally a summary slide of Chapter 8. I invite you to view this video or the videos by Dr. Cushman more than once, and good luck in all of your studies.